शुरू कर रहे हैं। अभी बज रहा है 1 दोपहर के। शुरू कर रहे हैं हम सेल लिमिट ऑफ लाइफ 25 मिनट। When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it that inanimate thing does not have, which a living thing has? This is the points of the basic unit of life. What is cell? Unicellular organism capable of independent existence and performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of all living organisms. After one Leibniz first saw and described a live cell, Robert Brown later discovered nucleus. The invention of the microscope and its improvement leading to electron microscope revealed all the structural details of the cell. Cell theory, 1838. Matthias Schleiden, a German botanist, examined large number of plants and observed that all plants are composed of different kinds of cells, and about the same time period as shown from 1939, a British zoologist studied different types of animals and reported that cells had a thin outer layer, which is today known as the plasma membrane. He also concluded, based on his studies on plant tissue, that the presence of cell wall is a unique character of the plant cells. On the basis of this, Schwann proposed the hypothesis that the body of animals and plants are composed of cells and products of cells. Schleiden and Schwann together formulated the cell theory. This theory, however, did not explain as to how new cells are formed. Rudolf Virchow, 1855, first explained that cells divide and new cells are formed from pre-existing cells. He modified the hypothesis of Schleiden and Schwann to give the cell theory a final shape. Cell theory as understood today is all living organisms are composed of cells and products of cells. All cells arise from pre-existing cells. An overview of cells. You have either observed the cells in your own field of human cheek. Cells under the microscope, let us recollect their structure. Okay. The cell of the human sheet have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane-bound structure called nucleus. This nucleus contains the chromosome, which in turn contains the genetic material. Contain the genetic material DNA. Cells that are membrane or nuclei are called eukaryotic, whereas cells that lack a membrane or nucleus are called prokaryotic. In prokaryotic and eukaryotic cells, same fluid matrix called cytoplasm is present. चलो अभी कागज चीज क्या है হুম? ছেড়ে দিলাম এবার। ছেড়ে দিলাম। অন্য জায়গায়। মজ্জা এসছে ক্ষমা চাইতে।
तो सामने जाएगा। मुझे क्षमा कर दो, गलती हो गई है। अरे भाई क्षमा बोला कौन मारे ना के।
Good, I like it. Beside nucleus, the eukaryotic cell have other membrane-bound distinct structure organelles like the endoplasmic reticulum, the Golgi complex, lysosome, mitochondria, microbodies, and matrix. The prokaryotic cell that lacks such membrane-bound organelles, ribosome, are non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast in plants and mitochondria and on the endoplasmic reticulum. Animal cell contain another non-membrane organelle called centrosome, which helps in cell division. Cell differ greatly in size. As for example, mycoplasma, the smallest cell 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in shape. They may be disc, polygonal, columnar, cuboid, and even irregular. The disc shape of the cell may vary with the function they perform. Prokaryotic cell are represented by bacteria, that is blue-green algae, mycoplasma and TP yellow. They are generally smaller and multiply more rapidly than eukaryotic cell. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, rod-like, orcus, spherical, Barbro, spiralum, spiral. The organization of the prokaryotic cell is fundamentally similar, even though prokaryotic exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane except in microplasma. All prokaryotes have a cell wall surrounding the cell membrane except in microplasma. Fluid matrix filling the cell is the cytoplasm. There is a no well-defined nucleus. The genetic material is basically naked, not enveloped by an nuclear membrane. In addition to the genomic DNA, the single chromosome circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids, and the plasmid DNA confers certain unique phenotypic characters of such bacteria. One such character is resistance to antibiotics, and one such antibiotics. In higher class, we will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotic cell except for ribosome. Prokaryotes has something unique in the form of inclusion. A specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryotes. They are essentially enfoldings of the cell membrane. Cell envelope and its modification. Most prokaryotic cells, particularly the bacterial cells, have a clinically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure. The outermost helix, followed by the cell wall, and then the distinct cell wall, and then distinct then plasma membrane. Although each layer of the envelope perform distinct function. They act together as a single protective unit. Bacteria can be classified into two groups on the basis of difference in the cell membrane and the manner in which they respond to staining procedure developed by Gram-Lewis. Those that take up the Gram stain are known as Gram-positive, and those that do not are called as Gram-negative. Glycocalyx differ in composition and thickness among different bacteria. It could be loose sheet called the slime layer in some, while in others it may be thick and tough called the capsule. The cell wall determines the shape of the cell and provides a strong structural support. Determines the shape of the cell and provides a strong structural support to prevent the bacterium from collapsing or bursting. The plasma membrane is selectively permeable. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of eukaryote. The special membranous structure is the mesosome, which is formed by the extensions of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamella. The special membranous structure is the mesosome, which is formed by the extensions of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamella. They help in cell wall formation, DNA replication, and distribution to daughter cells. They also help in respiration, secretion process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm. Cyanobacteriaতে membranous extension আছে যেটার নাম হচ্ছে chromatophores, which contain pigments. Bacterial cell wall may be motile or bacterial cells may be motile or non-motile. If motile, they have thin filamentous extensions from their cell wall called flagella. Bacteria show a range in number. Bacteria show range in number. Arrangement of flagella. Bacterial flagella composed of three parts: filament, hook, and basal body. The filament is the longest portion which extends from the cell. It extends from the cell surface to the outside. Besides flagella, pili and fimbriae are also surface structure of the bacteria, but do not play a role in the motility. The pili are elongated tubular structures made up of special protein. The fimbriae are small blister-like fibers sprouting out of the cell. In some bacteria, they are known to help to attach the bacteria to rocks and streams and attach to the host tissues. Ribosomes are the inclusion body. In prokaryotes, ribosomes are attached associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits, 50S and 30S, which when present together forms 70S. Prokaryotic ribosome. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single chain RNA to form chain called polysome or polyribosome. Ribosome or polysome transform the mRNA into protein. Inclusion body. Rhizome material prokaryotic cell are stored in the cytoplasm in the form of inclusion body. These are not bound by the membrane system and lie in the cytoplasm. Phosphate granules, amorphous granules, and glycogen granules, etc. Their granules are found in green, purple, and blue concentrate bacteria. Eukaryotic cell include all protist, plant, animals, and fungi. In eukaryotic cell, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. The eukaryotic cell possesses an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory structure and cytoskeletal structure. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different. as the former possess cell wall, plastids, and a large central vacuole, which are absent in animal cells. On the other hand, animal cells have centrioles, which are absent in almost all plant cells. Cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in the 1950s. Meanwhile, chemical studies of the cell membrane, especially in human red blood cells, enabled the scientists to deduce the possible structure of the plasma membrane. These studies showed that cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in bilayer. Also, the lipids are arranged within the membrane from polar head towards the outer side and the hydrophobic tails towards the inner side. This ensures that the non-polar tail of saturated hydrocarbon is protected from the aqueous environment. In addition to phospholipid, membrane also contains cholesterol. Later biochemical investigation clearly revealed that cell membranes also possess proteins and carbohydrates. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocyte has approximately 52% protein and 40% lipid, classified as integral and peripheral. Peripheral proteins lie on the surface of membrane, while the integral proteins are primarily or totally buried in the membrane. Peripheral proteins lie on the surface of the membrane, while the integral proteins are partially or totally buried in the membrane. An improved model of the structure of the cell membrane was proposed by Singer and Nicholson 1972, widely accepted as fluid mosaic model. According to this, the quasi-fluid nature of the lipid enables the lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of functions like cell growth, formation of intercellular junction, secretion, endocytosis, cell division. One of the most important functions of the plasma membrane is the transport of molecule across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and it is called passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient. From higher concentration up to the lower, water may also move to across the membrane from higher concentration to lower concentration. Water higher to lower osmosis. As the polar molecules cannot pass through the non-lipid bilayer, they require a carrier protein to the membrane. And to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient from lower to higher concentration. Such a transport is an energy-dependent process in which ATP is utilized. It's called active transport, sodium or potassium pump. one of the most important function of active transport. Carrier protein. Cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering of the plasma membrane of fungi and plants. Cell wall not only gives shape to the cell and protect the cell from mechanical damage and infection, it also helps in cell-to-cell interaction and provides barrier to undesirable macromolecules. Algae have cell wall made up of cellulose, galactans and mannans and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectins and proteins. The cell wall of a young plant cell, the primary wall, is capable of growth which gradually diminishes as the cell matures, and the secondary and the plant consists of cellulose, hemicellulose, pectins and proteins. Algae have cell wall made up of cellulose, galactans and mannans, and which gradually diminishes as the cell matures and the secondary wall is formed on the inner side of the cell. The middle lamella is a layer mainly of calcium pectate which holds or glues together different neighboring cells together. Their cell wall and middle lamella may be traversed by a plasmodesmata which connects the cytoplasm of the neighboring cells. So, the middle lamella traversed by plasmodesmata connects the neighboring cells together. Endomembrane system. While each of the membranous organelles is distinct in terms of its structure and function, many of these are considered together as the endomembrane system because their functions are coordinated. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. Since the function of the mitochondria, chloroplast, and peroxisomes are not coordinated with the above components, they are not considered as a part of endomembrane system. The endoplasmic reticulum. Electron microscope studies of eukaryotic cell reveals the presence of a network of a reticulum of tiny tubular structure scattered in the cytoplasm that is called endoplasmic reticulum. Hence, endoplasmic reticulum divides the intracellular space into two distinct compartments, luminal or extraluminal. Luminal inside ear and extraluminal cytoplasm compartment. The ear often shows ribosomes attached to their outer surface. The endoplasmic reticulum bearing ribosomes on their surface is called rough endoplasmic reticulum. In the absence of ribosome, they appear smooth and are called smooth endoplasmic reticulum. Rough endoplasmic reticulum is frequently observed in the cells actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site of synthesis of lipid. In animal cells, lipid like steroidal hormones are synthesized in smooth endoplasmic reticulum. 15 minutes break. 5 minutes break. Okay. Then we will start from Golgi apparatus. Okay, let's start Golgi apparatus. Emilio Golgi, 1898, first observed densely stained reticular structure near the nucleus. These were later named as Golgi bodies after him. They consist of many flat, disc-shaped structures or twisted cisternae of 0.5 to 1 micrometer diameter. These are stacked parallel to each other. Varied number of cristae are formed in the Golgi complex, concentrically arranged at the nucleus, and a distinct convex cis phase in the forming phase and the concave trans phase of the maturing phase. The cis and the trans phase of the organelle are entirely different but interconnected. The Golgi apparatus principally performs the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. Materials to be packed in the form of vesicles and are fused with the cis phase of the Golgi apparatus and move towards the maturing phase. This explains why Golgi apparatus remain in close contact with the endoplasmic reticulum. A number of proteins synthesized in ribosome on the endoplasmic reticulum are modified in the cristae cisternae of the Golgi apparatus before they are released from its trans phase. Golgi apparatus is an important site of formation of glycoproteins and glycolipid. Golgi apparatus क्या है एक important site है for the formation of glycoproteins and glycolipids. Lysosome. These are the membrane-bound vesicular structure formed by the process of packaging in the Golgi apparatus. Okay, so lysosomes, these are the membrane-bound vesicular structure formed by the process of packaging of Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes. Hydrolyzes, lipase, proteases, carbohydrate. Optimally active at the acidic pH. These enzymes are capable of digesting carbohydrate, protein, lipid, and nucleic acid. Vacuole. The vacuole is the membrane-bound space found in the cytoplasm. It contains water sap, excretory product, and the other materials not useful for the cell. The vacuole is a single bound by single membrane called tonoplast. In plant cell, the vacuoles can occupy up to 90% of the volume of the cell. In plants, the tonoplasts facilitate the transport of a number of ions or other material against concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protists, food vacuoles are formed by engulfing the food particles. Mitochondria, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending upon the physiological activity of the cell. In terms of shape and size also, considerable degree of variability is observed. Typically, it is sausage shape or cylindrical, having a diameter of 0.2 to to 1.0 micrometer, average is 0.5 and length is 1 to 4 micrometer. Each mitochondria is a double Each mitochondrion is a double membrane bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments, the inner compartment and the outer compartment. The inner compartment is filled with dense homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of enfoldings called the cristae towards the matrix. The cristae increases the surface area. The two membranes that have their own specific The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms the number of enfoldings called the cristae towards the matrix. The cristae increase the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP. Hence, they are called powerhouse of the cell. The matrix also possesses single circular DNA molecule, few RNA molecule, ribosome 70S, and the compounds required for the synthesis of protein. The mitochondria divide by fission. Plastids are found in all plant cells and in euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific color to the plant. Based on the type of pigment, plastid can be classified into chloroplast, chromoplast, leucoplast. Chloroplast contain chlorophyll and carotenoid pigments are responsible for trapping light energy essential for photosynthesis. In the chromoplast, the fat-soluble carotenoid pigments like carotene, xanthophyll and others are present. This is the part of the plant, yellow, orange or red color. The leucoplast and the colorless plastid, varied shapes and size. Amyloplast stores carbohydrate, starch, example potato. Allyloplast stores fats and oils, whereas the aleuro store protein.
हेलो एवरीवन, फर्स्ट ऑफ ऑल बहुत अच्छा लगा आप लोगों के पॉजिटिव फीडबैक देख के। मैंने देखा कैसे कैसे बच्चों का स्कोरिंग हुआ। 100 मार्क्स से 550 तक चला गया, 200 से 600 क्रॉस कर दिया। सिर्फ 7 डेज ऑफ 7 डेज हेल्थी चैलेंज। ये जो हेल्थी चैलेंज मैंने आपसे चीज़ें करवाई थी, यही सारी चीज़ें लिखवाई और मैंने ओपन चैलेंज दे रखा है। 2025 में जब पेपर होगा तुमको देखना है चैलेंज मैंने सारी चीज़ें कराई, एलिमेंट्स कराए, क्वेश्चंस कराए, मेरी लिस्ट ऑफ टॉपिक्स तुमसे करवाई, यहाँ से कितना पेपर आया। जो बच्चे लेट हैं लेट चैलेंज ज्वाइन किया है, लेट चैलेंज चालू किया था, कोई घबराने की जरूरत नहीं है। स्टेप में चैलेंज ठीक है। बाद में देखा तुमने वीडियो आई, वीडियो में तुमने देखा सबने अपने सपोज कर
Majority of the chloroplast of the green plants are found in the mesophyll cell of the leaf. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles are varying in length 5 to 10 micrometer and width 2 to 4 micrometer. Their numbers varies from 1 per cell of Chlamydomonas to green alga to 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplast are double membrane bound of the two. The inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called stroma. A number of organized flattened membranous sacs called the thylakoids are present in the stroma. Thylakoids are arranged in stack like the piles of coins of called grana, the internal thylakoids. In addition, there are flat membranous tubules called the stroma lamella connecting the thylakoids of the different grana. The membrane of the thylakoids encloses a space called lumen. The stroma of the chloroplast contain enzymes required for synthesis of the carbohydrate and protein. It also contains small double circular DNA molecules and ribosome. Chlorophyll pigments are present in the thylakoids. The ribosome of the chloroplast are smaller, 70S, and the cytoplasmic ribosome 80S. Thylakoids. The chloroplast are present in the mesophyll cell, lens-like shape, ribbon-like, 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplast are also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membranous sacs called the thylakoids are present in the stroma. Thylakoids are arranged in stack like the piles of coins called grana or the intergranal thylakoids. Double membrane bound structure of the two, the inner chloroplast membrane is relatively less permeable. The space limited by inner membrane called the stroma.
Stroma.
Number of organized flattened membranous sacs called thylakoid are present in the stroma. Thylakoids are present in the stroma. Thylakoids are arranged in stack like the piles coined called grana on the integral thylakoids. In addition, there are flat membranous tubules, flat membrane tubule called stroma lamella connecting the thylakoids of the different grana. Thylakoids arranged in stalk like coins piles called grana. The member of the thylakoid space called the lumen. The stroma of the chloroplast contain enzymes the stroma chloroplast contain enzymes required for the synthesis of carbohydrates and proteins. It requires small synthesis for carbohydrates and proteins. Double circular DNA molecules and ribosome. Chlorophyll pigments are present in thylakoid. The ribosome on the chloroplast are smaller, 70s, and cytoplasmic ribosome 80s. Ribosome are the glandular structure first observed under the microscope as dense particles called by George Palade 1953. They are composed of ribonucleic acid, RNA, and proteins and are not surrounded by any membrane. The eukaryotic ribosome are 80s while the prokaryotic ribosomes are 70s. Each ribosome has two subunits, larger and smaller. The two subunits of 80s ribosomes are 60s and 40s while that of 70s ribosome are 50s and 30s. The eukaryotic ribosome are the 80s while the prokaryotic ribosome are 70s. Each ribosome has two units, larger, small subunit. The two subunits of 80s are 60s and 40s while the 70 ribosomes are 50s and 30s. A stands for Swedborg's unit, stands for the sedimentation coefficient. It is a directly measure of density and size. Both 70s and 80s ribosomes are composed of two subunits. Cytoskeleton. An elaborate network of filamentous proteinaceous structure consists of microtubules, microfilaments, and the intermediate filaments present in the cytoplasm are collectively referred to as cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, and maintenance of the shape of the cell. Cilia and flagella. Cilia and flagella are hair-like outgrowths of the cell. Cilia and flagella are hair-like outgrowth of the cell. Cilia and flagella are the hair-like outgrowth like outgrowth of the cell. Cilia and flagella, which works. Cilia are small structure, work like oaks, causing the movement of either the cell or the surrounding the fluid. Flagella are comparatively longer and responsible for the cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of eukaryotic flagella. The electron microscope study of cilium and the flagellum show that they are covered with plasma membrane. Their core axoneme possesses a number of microtubules running parallel to the long axis. The axoneme usually has a 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Doublet. Such an array of axonal microtubules are referred to as 9+2 array. The central tubule are connected by the bridge and is also enclosed by a central sheet which is connected to one of the tubules of each peripheral doublets by radial spoke. Thus there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagella emerge from centriole-like structure called the basal body. 9+2 array axonal microtubules are connected by bridges is also enclosed by a central sheet which is connected to one of the tubules of each peripheral doublet by radial spoke. Centrosome and centriole. Centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar material. Both the centrioles in the centrosome lie perpendicular to each other, which is an organization like the cartwheel made up of 9 spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplet are also linked. The central part of the proximal region the central part of the proximal region the central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with the tubules of the peripheral triplets by radial spokes made up of protein. The centriole form the basal part of the body of cilia, flagella, and the spindle fibers that give rise to spindle apparatus during cell division of animal cells.
Very good. Do you want to see middle lamella and mid plasmodesmata? Color red is middle lamella and blue is
কয় বার নিয়েছিস?
শুরু
ও
कलमा पढ़ा रहे थे तो ये कलमा सुना है India has already updated
centriole centrosome is an organelle usually containing two cylindrical structure called centrioles. They are surrounded by amorphous pericentriolar materials. কি বলছিস? Oh. Pericentriolar materials. Both the centrioles in a centrosome lie perpendicular to each other, in which each has an organization like the cartwheel. Each has an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubular proteins. Each of the peripheral fibrils has a triplet. The adjacent triplets are also ringed. The central part of the proximal region is called hub and they are made up of spokes. The central forms the basal body of the cilia or flagella and spindle fibers that give rise to spindle apparatus during cell division of animal cells. Nucleus is a cell organelle first described by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dye was given the name chromatin by Fleming. The interface nucleus of a cell, when it is not dividing, has highly extended and elaborate nuclear protein fibers called chromatin, nuclear matrix, and one of the more spherical bodies called nucleoli. The interface nucleus of a cell, when it is not dividing, has a highly extended and elaborate nucleoprotein. The electron microscope revealed the nuclear envelope, which consists of two parallel membranes with a space between 10 to 15 nanometer, called the perinuclear space, forms a barrier between the materials present inside the nucleus and that of cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears an ribosome on it. At a number of places, the nuclear envelope is interrupted by minute pores, which are formed by the fusion of two membranes. These nuclear pores are on the passage which the movement of RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variation in number of nuclei are also frequently observed. The nuclear matrix or nucleoplasm contain nucleolus and chromatin. The nucleoli are spherical structures present in the nucleoplasm. The content of the nucleus is continuous with the rest of the nucleoplasm, as it is not a membrane-bound structure. It is a site of active ribosomal RNA synthesis. Larger or more numerous nuclei are present in cells actively carrying out protein synthesis. The nuclear matrix contains nucleoplasm, contains nucleolus and chromatin. The nucleoli are spherical structures present in the nucleoplasm. It is a site of active ribosomal RNA synthesis. You may recall that interface nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin, but during different stages of cell division, cells show structured chromosome in which the nucleus chromatin contains DNA and some basic proteins called histones, some non-histone proteins, and also RNA. A single human cell has approximately 2-meter long thread of DNA distributed among its 46 chromosomes. We will study the details of DNA packaging in the form of a chromosome in class 12. Every chromosome essentially has a primary constriction or the centromere, on the sides of which disc-shaped structures called kinetochores are present. Centromeres hold two chromatids of a chromosome. Based on the position of the centromere, the chromosome can be classified into 4 types. The metacentric chromosome has middle centromere forming 2 equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. In case of acrocentric, the metacentric equal arms, submetacentric one from the middle of the chromosome remaining one shorter and one longer, acrocentric one close to the end forming one extremely short and very long arm. Whereas the telocentric has a terminal centromere. Sometimes few chromosomes have non-staining secondary constrictions. Non-staining secondary constriction at a constant location. This gives a small appearance of fragment called satellite. Microbodies, many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells. Done. So we have a break time for lunch. After lunch, we will start again. Bye bye.
And we will distribute the primary tissues. The primary tissues में apical apical मतलब जो roots हैं, जो tips of roots and shoots में रहते हैं, उसे apical कहते हैं. और apical मेरिस्टेम and apical मेरिस्टेम जो कुछ बचा रहेगा तो उसमें apical कहते हैं. Both ही है हमारा primary plant को primary formation of body. ठीक है. अब हम इसका एग्जांपल क्या सेकेंडरी मेरिस्टेम का GS स्क्वायर. Secondary meristem related and secondary meristem. So examples of fascicular, vascular cambium, interfascicular cambium, and cord cambium. Fascicular, vascular cambium, interfascicular cambium, and cord cambium. Fascicular, vascular cambium, interfascicular cambium, and cord cambium. এবার আমরা করবো কি করবো primary meristem করবো secondary meristem. দুটোতে meristem করবো আর দুটোকে divide করছি. পরে আসছে major regions of roots and shoots and plants পরে আসছে কিন্তু দুটো ডিভাইড তো করছে পার্মানেন্টের মতো না বসে ঘুরে ঘুরবো. Permanent mature cells কি আছে newly formed cells become structurally and functionally specialized. সেগুলো ফর্ম হয় হঠাৎ করে কিছু কিছু তাদের মধ্যে স্ট্রাকচার অ্যান্ড ফাংশনালি প্রচন্ড স্পেশালাইজড হয়ে যাওয়ার ফলে কি হয় lose the ability to divide. Dividing ability. The third cells are known as permanent or mature cell. Which is permanent or mature cell? Specific region of apical meristem during formation of umbilical plant body produce. Specific region of apical meristem during formation of plant body produce. Produce the body, dermal, ground, and vascular tissues. Normal tissues, dermal tissues, ground tissues, and vascular tissues.